Yeah, before we start off and kick off with the presentations of today, I want to just draw your attention shortly on the online presence of Euchis. We have a wonderful homepage which is maintained by Stefan Hoffmann, where you can find all the necessary information, all online resources, but also information about the webinar and many more. Additionally to the email address that most of you or the members of Euchis might be familiar with, which is the one of Martin Peter. We have an additional email address now, which is euchis.online@gmail.com. So if you're interested or if you have any requests regarding Euchis, or if you're interested to contribute to this webinar series, please just drop us an email and we try to answer in time. You can also communicate with us via Twitter. You can find us at Euchis_Chitin. And the very nice feature that I really enjoy is the YouTube channel. We have the webinars. If our presenters agree, we're gonna ask them before or afterwards. We're gonna put the presentations online of the webinars, so you can watch it also offline anytime you want. For example, the first two webinars are online already, including the presentation of Bruno Mörschbacher, which is really a perfect overview about chitin and chitosan sciences. Yeah, and with that, there is also the possibility to join Euchis as a member. It's a really fruitful network association that I enjoy as a young scientist, but I think it's also very fruitful for established scientists. So if you're interested in joining Euchis as a member, you can find all the necessary information on the website euchis.org. There is already a handle set up for that. So with that, Thomas, I would like to start with you since you're now here. So Stefan, if that's okay for you, we're gonna put you a second. And Thomas, if you want to try to, or I'm not sure, since you just recently joined, I still have to co-host you.
Okay.
So now it should work. If you can share your presentation with us.
Just a moment. Now it should work. And if I start the presentation, what do you see?
So can you go on?
Is it okay?
Yeah, perfect.
Is it okay?
We can see it all works. Okay, perfect. So with that, Thomas, I would like to introduce you shortly. Thomas studied chemistry and obtained his PhD at the Institute of Bioprocess Engineering at the University of Kaiserslautern, thereby working on the extraction and purification of heteropolysaccharides from brown algae. In 2012, Thomas started as a research scientist at the Fraunhofer Institute for Interfacial Engineering and Biotechnology, IGP. And since 2016, he's an assistant head of the working group Bioprocess Development there. The research topic of Thomas includes the extraction and purification of natural polysaccharides from renewable resources, which also includes chitin, the topic of today, and also the chemical and biotechnological modification of those polysaccharides. In his research activities, he's working with several companies and research institutes with the aim to foster the application of renewable resources in industry. And we are very happy that you're gonna join us today, Thomas, and looking forward to your presentation.
Yeah, thank you for the kind invitation and the kind introduction. So what I'm talking about is a chitin extraction and chitosan production from non-marine sources. And if I talk about non-marine sources, I focus on side or waste streams of the insect protein production. And as you can see here on the right side, there's a close-up image. Does look a little bit nasty. There's one on the one side, on the left side, these are larval exoskeletons. On the right side, these are pupal exuvia. So my agenda is as follows. So first I give you an overview about different chitin sources. Then I will follow up with the development of analytical methods, which is really important to evaluate the chitin purification, which is followed afterwards. And as you maybe know from other sources, we have different process steps like demineralization, deproteinization, and bleaching. And of course, the chitin chitosan production follows. And I want to give you one example. You already maybe know this is a chitosan as used as a sizing agent in weaving. That means in textile processing. And as last step, I want to give you an outlook and talk about further current activities. So there are three main sources available for the chitin in the nature. On one hand, we got the fungi with chitin and chitosan in the cell wall embedded in beta-glucan complexes. And as you maybe know, there is also there is the phyla Arthropoda with the subphylum crustaceans, which is the common source for chitin. And what I want to talk about today is the hexapoda, which are also a source of chitin, where the chitin is mainly embedded in the exoskeletons, additionally consisting of proteins and minerals. And in the case of hexapoda, catecholamines. In the case of crustaceans, these are additionally carotenes. So which is the best source? It is not so easy to answer that question, but as always, chemists say it depends on. And I want to present you a comparison. So on one hand, the fungi are a vegan source in contrast to crustaceans and insects, which are animal origin. The chitin is embedded in the cell wall of fungi, whereas it is embedded in the exoskeletons of the crustaceans and the insects, as we have already mentioned one slide before. And the fungi, it is integrated or complexed in a matrix with proteins and glucans, whereby it is embedded in the proteins and minerals, or in the presence of minerals in crustaceans and insects. And in crustaceans, there are additionally carotenes. In insects, there are additionally catechols. And the chitin can be derived in general from waste streams, like in the fungi, from the citric acid production. So at the moment, we got about 50 kilograms of Aspergillus niger from the citric acid production, which we're using to purify the chitin. But you could also take some waste streams of the mushroom cultivation. Yeah, for the crustaceans, you already know, which is common, that we can purify chitin from marine fishery waste, and insects from the insect chitin from the protein production. So the potential, and I need to emphasize that the potential annual chitin production from these wastes exceeds for the fungi more than 200 kilotons worldwide. For the crustaceans, I made some calculations. I ended up in between 0.6 and 2 million of tons, which can be obtained from these marine fishery waste. And what is a little bit disgusting or disappointing is there are only 15 tons currently available from the insect protein production. But I need to emphasize that is only a Europe number. So the question arises, of course, why using insect-based chitin? So insect production facilities are launching worldwide, and we saw in preliminary investigations that there are some side streams of the insect cultivation which are rich in chitin. And if you have a look on different literature, you will see that we can estimate a volume of about 15 kilotons in 2030 in Europe of chitin derived from insect protein production. And this means it's more than 1,000-fold. So that means also for us, it is a promising prospective delocalized chitin source, not a current chitin source. So what do we do with these insects or insect cultivation? So first we need to have a look at the life cycle of the Hermetia illucens, which is also named the black soldier fly, which is currently used to obtain this protein. First, we got our eggs, which develop for 6 to 8 days, and one fly or one adult lays about 500 to 1,000 eggs. They develop to larvae, which incubate or grow for 6 to 12 days with organic residues. Organic residues is, for example, spent grain or any fruit production wastes and so on. And what you can isolate from it first, it was the lipids. So they press the larvae out. So you get the lipids from it. Then you get the organic fertilizer, which is also built by frass, which was separated before the larvae was further processed. And in the end, we got the the proteins, which is the residue of the larvae processing. But a part of these larvae develop to prepupae in several days. And during this transformation, they shed their skins about 6 times, which comes to the batch of larval exoskeletons. And a further the further development to pupae, which takes about 10 to 14 days, and they do not uptake any feed, then they shed also their skin to develop to the black soldier, to the adult larvae, and we got the pupal exuvia. So then at the beginning, our question was, or the challenge was to determine the chitin content. If you only have to, if you know the chitin content, you could only evaluate your chitin purification. And the challenges to meet here are, on one hand, the sclerotization. That means this is a process which involves the cross-linking of chitin, protein, and acts and uses catechol amine as a cross-linker. That means the skin, the exoskeleton gets harder, it gets darker, water was released, and so on. And that also means there is a low accessibility for this chitin, for example, for enzyme activity or for anything else. So what we first have done is then to try to use, to try to detect glucosamine after total hydrolysis. Yeah, but there was a special equipment needed or required. And what is also important, you need to determine the suitable conditions. And that means you should hydrolyze the chitin to end up in glucosamine. But on the other hand, you need to, there is the threat to glucosamine degradation. So that means you could not determine the glucosamine anymore. So we decided to determine acetate groups after total hydrolysis. And we, at this moment, it was several years ago, we stated that we got an acetylation degree of about 90%. Currently, we know that the acetylation degree in the insect chitin is about 95 to 96%. So we correlated the acetate in the supernatant to the chitin in the end. And what we also have done, so because you're also needing hydrolysis and HPLC measurement, what we also have done is to compare it with the gravimetric method. We adapted from the feedstuff characterization. Our intention was first to measure the fiber content, and we saw there's a chitin analog. On the other hand, we wanted to measure the lignin content, the ADL, to consider for the catechol amine amount. And I want to explain that to you in detail, these ADF minus ADL method, how we name it. So on one hand, you got the feedstuff with the cellulose, the lignin, hemicellulose, starch, sugar acid, and ash. And we said that is corresponding to the insect, which include carbohydrates, proteins, lipids, chitin, ash, and catechol amine. Then we degraded with a acid detergent and heat it up. So there is a gravimetric determination in the end for the feedstuff, which includes cellulose and lignin. The insect biomass includes chitin, insoluble proteins, and catechol amine. So, and if you can see it here on the left side, the chitin content of the ADF, that means the first process step, the first gravimetric determination, overestimates the chitin content. If you can see, there is a small deviation to the acetyl group determination or the acetate group determination, which we think is the most accurate, and we also determined that statistically that is the most accurate method. So, but the next step was then to hydrolyze these fiber, the retentate, with sulfuric acid, with concentrated sulfuric acid, and this results for the case, or in the case of the feedstuff, in the lignin, or in the case of the insects, for the acid-resistant catechol amine. And you see here, there's a small ADL arrow, and if you subtract these ADL from the ADF, you end up in the acetyl group determination. That means you got now a tool which is also accurate but does not need really that much equipment to measure the chitin content. And this is also something we already published several years ago. So according to statistical analysis, you see here on the right side, we measured with the different methods. We measured the chitin content, larval exoskeletons, pupal exuvia, and also as a benchmark, we used shrimp chitin. And we also saw that the ADF alone is not suitable. But what we also can state is that the ADF minus ADL method gives comparable results to the acetyl, to the acetate method. But to be honest, if you want to measure anything with these ADF minus ADL method and you got something like cellulose inside, it disturbs your analysis. It's the same also for the acetate group determination. If you got some hemicellulose inside the sample, it will disturb your method and will also give you wrong values. You always should consider that. Okay, we see here that the larval exoskeletons and the pupal exuvia have a high chitin content. For larval exoskeletons up to 40% chitin, and for the pupal exuvia up to 25% of chitin. Now I will come to the purification from pupal exuvia and larval exoskeletons. And the first thing is, it is like crab shell or the crab chitin purification. We determined a high ash content in the side stream, so up to 25%. And these minerals we identified with ICP-OAS, and we saw that calcium is the main metal ion, which is inside of the in the insect residues, and calcium carbonate is the main salt with 65 to 80 milligrams per kilogram. But if this is a carbonate salt, you could also solubilize it with acids. So what we have done is then to compare different acids for salt solubilization, on one hand at 40 degrees and on the other hand at 70 degrees. And we saw that with formic acid and HCl, more than 90% of the minerals were removed with both. But in common, the people from industry, they don't like to use hydrochloric acid. So in common, it is better for us to make an industrial process if you use formic acid, which is not that strong as hydrochloric acid. But on the other hand, with other sources, we also have seen that hydrochloric acid is much more efficient. I think this is also it's due to the strength of the acid. Okay, second step, as you know, we wanted to deproteinize our source, our biomass. And we had the first challenge, which took us a long time. So because the protein measurement was a real challenge due to the presence of catecholamines. And if you maybe know that if you use some ultraviolet or spectrophotometry or anything else like Bradford assay or anything like that, these get disturbed or distorted by the presence of these catecholamines because they are also redox active. And what we have done is then to solubilize these proteins, to precipitate, to purify, and then to measure them. Or what we also do now at the moment to measure the fiber content, and we know how much nitrogen is into this fiber, and then we subtract this nitrogen content included in the fiber from the total nitrogen content, and know which protein content we have. So we screened several commercial enzymes, which was not really successful. So alkylase application gave partially deproteinization, but it was not really successful. There was a microbial activity, so we determined one microorganism which was really successful concerning demineralization and deproteinization, and that was really promising. But I think we need to put much more effort in that to come or to correlate that or to compare it to the solely chemical deproteinization. And these chemical deproteinization we evaluated or assessed with design of experiments approach, as it is common. The more drastic the conditions, like higher sodium hydroxide concentration or higher temperature, the better the deproteinization, and our protein removal efficiency exceeded up to 95% in this case. And what was also benefit is that the catecholamines were also removed as well, but not that much, as you can see here on the right side. Here you see the pupil exuvia at the beginning, at the picture above, and in the end you see that there is a slightly brighter than before, but there are some catecholamines left. And this is the reason why we followed with the bleaching step. And what we have done is then to compare several bleaching agents we identified in the literature. What we saw is that sodium hypochlorite performs the best, and we made some kind of quantitative evaluation via colorimetry. We used our biomass, pressed it together, and put it in an equipment. It's called CL Lab. If you want to know more about this, you can also ask me. And then it is possible to make a quantitative evaluation. We saw that the highest effect gave sodium hypochlorite, but this is not really sustainable. But what we saw is also more than a sustainable alternative is then to use hydrogen peroxide. And what I haven't told you before is that maybe at the beginning for the pupil exuvia, we got a ADL content of about 12 or 30%. That means it's really dark material. And during these bleaching with sodium hypochlorite or H2O2, we achieved a reduction of these ADL content of the coloration of more than 8%. And these are the current results of the chitin. We got a chitin content of about 90%. Before I published now something with about 85%. So we improved that. We got an acetylation degree of 96% with a solid state NMR. We got a crystallinity of about 74%. And if you want to ask if this chitin content is really, if we can increase that, I do not really know. This is at the moment, this is some kind of a magical threshold, but we will see what happens. So if you're interested in some further literature, or if you want to ask me or my boss, here's contact and literature. Don't hesitate to ask me. And I want to say thanks for BMBF for funding for the project implementation of the Chitotex. I want to thank Fraunhofer for funding. It's a new project which started at the beginning of this year, the treatment of complex process wastewaters with biofunctional bio-based flocculants, and to the University of Stuttgart in the person of Linus Stegbauer and Niels von Segern, which was a good collaboration partner of us, and to all the people which are involved here in this research. So thank you for your attention, and I think you got some questions.
Yeah, thank you so much, Thomas, for this contribution. It was really exciting to see your activities at the Fraunhofer Institute there. Yeah, just forwarding the quest for questions. Are there any questions out there? Please raise your hand or just like shout them in.
May I have a question?
Of course. Okay,
thank you. Thank you very much for the presentation. You just mentioned very briefly the use of ionic liquids.
Yeah.
If I remember well, it was for getting chitosan, okay, from chitin, if I remember well. Can you please give more details on the possibilities and also of possible failure of this method? Thank you.
On the one hand, I also remember that our collaboration partners, they used ionic liquids not to convert it to chitosan. They used the chitin directly to make a fiber which is blended with cellulose, because chitin itself has not really good chemical physical properties to produce a fiber or produce a yarn. So there is a need to blend it with other polysaccharides. But I also remember that some people I know, they use these ionic liquids for purification, because afterwards, if you use these ionic liquids, you can precipitate, I think you can precipitate all other contaminants out, and the chitin is a residue, is purified residue of this process.
Okay, thank you. This was the reason why, okay, the use is interesting in some way. Okay, thank you very much.
You're welcome.
Okay, thank you, Carlo. The next question was from Martin.
Yes, can you hear me?
Yeah, we can
yes.
hear you.
Yeah. Concerning the catecholamines, I think the picture that you show is too simple. The in the sclerotized cuticle, at least, of the pupil exuvia, these are polymers. You can compare it a little bit like cellulose and lignin. And actually, we have done many years ago a lot of studies on the mechanism of sclerotization. So there is an oxidative process, and this is it gives a polymer action. So it is like a plasticide, the role of the catecholamines. Therefore, it will be very difficult to remove them because the interactions are not only covalent, but they are also on hydrophobicity and hydrogen bonding activities. So actually in cellulose production, you have different processes to remove the lignin, which is the sulfur process and so on. I don't know whether this would be applicable also to isolation of chitin. Okay. And the second point or question I have is when you determine the acetyl groups, the catecholamines or the polymer which is generated from them contain also acetyl groups. So because it's N-acetyl dopamine, and this could also give analytical results which must be considered.
Okay, thanks. So where shall I start? A lot of questions, but thank you for your comment. So on one hand, you're absolutely right. This was a simplified visualization of the current state in the insect cuticles, of course. And then we have already tried to make an organosoft process, which was already or currently used for the lignin removal from cellulose process, and it was not really successful. We only made 2 or 3 fractionations. It was not really successful, but we already had the same already had the same idea to do this. It wasn't really. And on the other hand, you're also right, the N-acetyl dopamine will also cleave during the hydrolysis, but I think this is not that much, and of course it will disturb our measurement. But on the other hand, if you really got hemicellulose inside, so some people come to me from the industry and they say, okay, how could we measure the chitin content within frost? That means within the insect cuticles with the feed stuff and so on. And sometimes I say, well, that's really hard to do that with the ADF-ADL method because of the hemicellulose inside, and because also of the cellulose inside. But we are currently busy to adapt and optimize the method, because the benefit of this method is that it could be done with equipment which is not sophisticated.
Did you try peroxidases?
Yeah.
Like the fungal enzymes?
Maybe you haven't. It was just a brief overview about the bleaching agents, because on the very right side you saw we used lacase. We used lacase with mediator and without mediator, and it was really hard. It was not really successful, and I think this is really a huge challenge, because these catecholamines mainly perform these covalent bonds, and these covalent bonds is also not so easy to remove with bleaching agents and so on.
Okay, thank you.
So you're welcome.
Thank you so much. One last question from Vincent.
Hi, Thomas.
Hi,
very nice talk. The work that you described is really, really important. Just a quick comment before I get to my question is that we have also tried things like steam explosion in the Gitotex project. You may remember something that you do for lignocellulose processing, which also didn't work very well. And we in my lab, we actually say as a joke that it's much easier to get cellulose out of a tree than it is to get chitin out of an insect shell. But that's for later discussion. But my question, did you, because you've done all this work, did you make any economic considerations about the eventual production costs of this chitin vis-a-vis compared with, for example, crustacean chitin? You have any idea about that currently?
No, haven't done that yet, but because a lot of companies are interested in that, so you maybe remember it from Protex. They see this is very promising for them, and we need to differentiate between larval exoskeletons and pupal exuvia. The larval exoskeletons, there are in in the presence of RAS. That means it is really hard to separate them from other contaminants. The pupil exuvie, they told me from Protex, pupil exuvie, it's no problem to get them in a purified form. But you maybe also remember it's really hard to get any chitin from that. It's much easier to get chitin from Lavalex's gland. That's the problem at the moment. But yeah, if I would say it's more economic, no. So I think from crab shell chitin to purify and to isolate chitin from crab shells is at the moment much more economic because you don't have that much covalent bonds or cross-linkings.
Yeah. Smart to do, in other words. Yeah, we would have some ideas how to tackle that. Yeah, but okay. Thanks.
You're welcome.
Okay, thank you so much, Vincent. And thanks again, Thomas, for this really interesting presentation. Thanks for joining us today. It was really exciting to hear all about that. In the interest of time, I would like to switch to the second part of today, which is the presentation of Stefan Cord-Landwehr. Stefan, maybe we can set up your presentation very quickly before I'm gonna introduce you. Still don't hear you, Stefan. Maybe, or do you have
you have to activate it again.
Oh, okay. So are you? Does it work now? Perfect.
No.
Perfect. Yeah, I'm really looking forward to your presentation, and it's a pleasure to introduce you, Stefan. Stefan studied biotechnology at the University of Münster, at which he also obtained his PhD in the lab of Bruno Mörschbacher. His thesis focused on the quantitative mass spectrometry for analyzing chitosan modifying enzymes and how they define the bioactivities of chitosans. And from your PhD thesis, I think there emerged several really crucial publications that made many of us aware of the research activities and also your PhD thesis. And I think for your thesis, you were also awarded the Braconnot Prize by the European Chitin Society in 2017. So congrats to that too. And Stefan is currently postdocing in Bruno's lab still at the Institute at Biology and Biotechnology of Plants at the University of Münster. And I'm really looking forward to hear about your research and progress there.
Thank you, Gregor, for the nice introduction. And my presentation will more focus on the analysis of chitosan. So, and here mainly how enzymes can help to analyze more complex chitosans. Like in the last presentation, a lot of was talked about chitin, but we focus more in our group on the chitosan and especially the partially acetylated chitosan that consists of glucanol and glucan units. The reason why we are focused more on these chitosan is that there are a lot of different bioactivities of these chitosans, and these bioactivities depend on the different parameters of the chitosan. So it's either the degree of polymerization that can influence the bioactivity, like you can see here that like the medium range chitosans have antimicrobial activities, and also the degree or fraction of acetylation has an influence in the bioactivity. For example, this medium ranged chitosans according to DA and high DP are very helpful in plant stuntioning activities. So these are two very crucial parameters for chitosans, and they are of course analyzed since several years. In the last years, the chitosan you can get on the market are much better described, and you can more and more trust these values. But there's even a third parameter that might be very interesting in the chitosan research and also for the bioactivity. This is the pattern of acetylation. And the pattern of acetylation means how these two acetylated and deacylated groups are distributed in the chitosan. And we believe that also this pattern has a strong influence in the bioactivity. So therefore, I have put in methods to analyze all of these three parameters. I think at the moment, all chitosan you can get on the market is starting from chitin, mainly shrimp chitin, but maybe in the future also from other sources like you saw in the last presentation, like from insects or maybe also fungi. You use very harsh chemical conditions like sodium hydroxide to get chitosan. You can even further degrade them to get chitosan oligomers. At the moment, all of these chitosans which you can get have a maybe more or less defined DP and DA, depends on the method you're using, but the pattern of acetylation is all random for them. But instead of using chemicals, you can also use enzymes, for example, chitin deacetylases. And these chitin deacetylases can convert chitin, as the name says, into chitosan. Unfortunately, these enzymes are not really active on the pure chitin because it's too crystalline. So at the moment, if you want to use this enzyme, you have to go this way, first using a sodium hydroxide pretreatment, and then you have a partially deacetylated chitosan, and then you can use an enzyme and further deacetylated ones. Since sometimes there's also a new way we also try in our lab, we start with a fully deacetylated polyglucosamine, using also CDAs, using high acetate concentrations, and by this you're also able to get chitosans. And now the question is, the question is, how does these chitosans look like? And for that, I will tell you in the next few minutes somehow about enzymes that might be helpful in the analytical methods for these chitosans. So one very interesting enzyme is, of course, it's a chitosanase, and the other one is a chitinase, because these enzymes can degrade the polymer into oligomeric parts. And the question is, why do we need these enzymes? Is most of our analysis we are doing with mass spectrometry, and mass spectrometry is good for small samples, like for oligosaccharides, but you cannot use it for polymers. But it's important, if you're using these enzymes, you don't want to lose the information about your chitosan, especially the degree of acetylation, also the pattern of acetylation. But to have this information, you have to know first how these enzymes can cut the polymer into oligomeric part. Here it's shown in a simplified way that the chitosanase can cleave between two deacetylated units, and chitinase can cleave between two acetylated units. But I think we all know from literature and from our experiments that it's not really true. Enzymes have different specificities, and they're not only true for these two sugar units, there are also the sugar units that are not binding so close to the active site has an influence. And how to analyze these enzymes, you can see here in this simplified figure, if you degrade your chitosan polymer into oligomeric parts, specific positions in the new formed oligomers are connected to specific subsites of the enzymes. Like the new formed reducing end was bound at the -1 subsite of the enzyme, and the new formed non-reducing end of the oligomer is bound at the +1 subsite of the enzyme, and so on. So for that, we used this method that was published a few years ago, where you could do a quantitative analysis of all these different chitosan oligomers, which you can produce by enzymatic hydrolysis of chitosan polymers using chitinases, and doing a few chemical modifications to achieve a quantitative analysis, and that allowed us to quantitative analyze the chitosan oligomers in a DP range of 1 to 6, according to the degree of polymerization, degree of acetylation, or the pattern of acetylation. And then this method we used to analyze first how these enzymes work. We started with one enzyme that was very well described in the group from Ising. It's a Chi-B from the Hermetia, to be sure that our method works as good as their methods, and that you got the same results. And by using our method, we could show, as expected, for this enzyme that belongs to a GH18 family, that this enzyme have to have an A at the -1 subsite, so that all oligomers which you get have an acetylated unit at the reducing end. The other subsites are not so 100% specific, but you could see some preferences that, like, the enzyme prefers to have an A at the -1 subsite, and so on. Then we use this method to for a lot of other enzymes. On the one hand, to understand how these enzymes work, but then on the other hand, to have much more tools for our analysis we plan with these enzymes. Another example is this chitinase glycoderma virulence. where we did the same kind of analysis, degraded different chitosans with different DAs, checked the substrates at different time points, and as a result, we got is almost the same like for KB. You have to have an A at the -1 subsite. Then there are different preferences for acetylated or deacylated units at the different subsites. So these enzymes has a higher preference for acetylated units than KB. But in total, it does not differ so much from the enzyme. We performed this analysis for even more enzymes, including chitosanases, and as you can see here, for different GH8 families, like GH8 or GH46. And we could show that these enzymes have different absolute specificities or also preferences, like this enzyme from CSN 1N74. Requires only a deacylated unit at the -2 subsite. The other 3 chitosanases, they can cleave the chitosan only after 2 deacylated units. Then we even further use this method for one very special enzyme, that is the chitinosanase from Alternaria alternata. And this enzyme is not called chitinase or chitosanase because, except to all other enzymes I showed you before, this requires an acetylated unit at the -1, like the GH18 chitinases, but it also requires an D at the -2 subsite, like the chitosanases. So it's somehow a combination of chitinases and chitosanases, but it has to have this pattern, and then the enzyme is able to cut it. So by doing that, we had a library of different enzymes that can cleave chitin or chitosan in a different way. You get different products, you have different specificities, and some are more specific, like the chitinosanase. Some are not so specific, like KB or some chitosanases, but all have their advantages and disadvantages. So then the next question was, can we use these enzymes to analyze the degree of acetylation and mainly the pattern of acetylation, because this is a very interesting parameter, but very difficult to analyze parameter. Because the idea is that both of these parameters can analyze it. And there are some methods known already how to analyze these parameters. A lot of them are from Kiel-Warum. They are very good methods. Maybe the only disadvantage is that you need, according to the research we are doing, a lot of material. Sometimes 10 milligrams are not so much, but sometimes 10 milligrams of chitosan is a lot. If you have this amount of chitosan, you can easily do the HNMR method, and you can easily determine the degree of acetylation. But we wanted to try using less material, so go to the microgram scale. And for that, we wanted to try if it's possible to combine the enzymes and mass spectrometry to also determine the degree of acetylation. So here our idea was, and this was also mainly done by these 2 guys, Anna and Jasper, who did also their PhD in Bruno's group in the last years, using 2 enzymes like a chitinase and a chitosanase to get a lot of different chitosan oligomers, and then using the information from these chitosan oligomers to determine the degree of acetylation. So to have a less complex sample, we decided to using enzymes that are unspecific, because this allows us to get small products, but not too small products. Like maybe you remember from the last presentation, if you have monosaccharides, especially glucosamine, it can easily react with other compounds and it's not so stable. So for that, we focus on enzymes that were able to degrade the chitosan polymer in a range between and to generate products somewhere between DP2 and 5, maybe as you can see here. But then still you have a lot of complexity in your sample. You have different DPs and different DAs. And to reduce this, we again included an isotopic labeling step, as you can see here, using acetic anhydride containing deuterium. And by doing this chemical modification, you ended up with fully acetylated chitin oligomers, but because of the deuterium inside the new acetyl group, you can distinguish the new from the old acetyl groups. Then we performed an LCMS analysis, and we could use a relative intensities in each DP to determine the degree of acetylation. So this is here on the left hand side, you can see how these results look. You get the different intensities for the different chitosan oligomers ranging from DP2 to DP5. You don't have any larger oligomers because of these two enzymes, so they're able to break everything into something between DP2 and DP5. Because of the recirculation, it's now possible to compare the intensities in one DP block. And by using this information, we were able to determine the degree of acetylation of several chitosans, as you can see here in this table. And we compared these results using, on the one hand, HNMR, that is the gold standard, the most precise method that's known, and also the titration method that's used in commercial chitosans because it's a method where you don't need very expensive equipment for. But as you can see here, there's not a huge difference between the DA determined using the ORMS method, and also not a huge difference except for one sample if you compare the results with the titration method. And the huge advantage of this method is that you need something between 10 and 30 microgram of chitosan polymer. So this method is very good if you have only very small amounts of material, but of course it's much more complex than using the HNMR method. Then the second question is now, as you should, maybe as you remember from the first slides, if you're now using enzymes to produce chitosan, or maybe if you get this chitosan already from natural sources like from fungi, do these chitosans have the same random DP of acetylation like the one you get if you're using chemical production rate, or do we get some non-random pattern of acetylation? Again, there was a method published by Kivarum 30 years ago that enabled us to determine somehow the pattern of acetylation. You could determine how often you get this 4 different dietes, the DA, DD, AA, and AD diet. I have a deeper look into the C5 region of a 13C NMR measurement of your sample. But the disadvantage is that you need about 100 milligram of this chitosan polymer to get good results. And the problem is, for our experiments, where we're using with our enzymes, we don't have such a huge amount of chitosan polymer. Maybe we have a few milligrams, but sometimes only some microgram amount. So there, again, we had the idea, can we use again enzymes to, to work with less material, again, to be in the microgram scale. But now, if you want to analyze the pattern of acetylation, it's important to not degrade the enzyme to the chitosan not too strong, and to somehow keep the information of the pattern inside. And therefore, again, many number Anna and Jasper, idea was to use a chitinosanase because we know that this enzyme has a very, very interesting specificity. So the enzyme is only able to cleave behind the DA sequence. So that means that the A's as well as D blocks are not destroyed if you're using these enzymes. And maybe if you see it here, if you have these 3 different patterns, like a random or more regular or blockwise pattern, you end up with different products. This means if you're using this enzymes and have different pattern of acetylation, you will get different products. So it means we have different fingerprints. So therefore, we produced different chitosans chemically or using different chitin destylases, checked them with 13C NMR if there are some differences in this diet formation. And what you can see, maybe if you compare the DD to the AA block, there is sometimes a huge difference in these 2 peaks. For example, in chitosan 3, this AA peak is much higher than in the other chitosans. And by doing some further analysis, you could estimate that this chitosan 1 and 2 have more random pattern of acetylation. This chitosan 3 has more blockwise pattern of acetylation, and the chitosan 4 has a more regular pattern of acetylation. And now by having these different chitosans, we could check if our idea to use the chitinosanase to determine the pattern of acetylation, or to get at least an idea about the pattern of acetylation, is working. So for that, we again use the enzyme, got a lot of different products, used LCMS measurements, and analyzed all of these different products. Then we had a semi-quantitative LCMS measurement, because we didn't include any internal external standards because there are too many samples. But if you have so many different products, you could do principal component analysis. And here you could already see, that the enzymatically produced chitosans, it's chitosan 1, 2, and 3, differ from the chemically produced ones. So this just shows you that they are somehow different. But more interesting is what you can see here on the right-hand side, is we were also able to determine or to calculate the average A block size and the average D block size. It's not the 100% correct values, because we are losing the very large oligomers. But what you can see here, for example, is that this chitosan 1 has a much larger average A block size than they expected for this degree of acetylation. All of them have a degree of around 30 or FA of 0.3. So, but here you have much more A blocks. And if you then compare this one with the results from the 13C NMR, that's this one here, there we also got the information that this chitosan has much more AA dietes in the polymer than you would expect for random pattern acetylation. So by using this combination of chitinosanase and mass spectrometry and this bioinformatic analysis, we have now at least an idea how these chitosans look like. Of course, it's not 100% good description of the chitosan, but we can get an idea if the A or D block size is different from the one you expect for a random pattern of acetylation. Of course, a lot of experiments have to be done here to get a very good picture of the pattern of acetylation, but at least now this method allows us to do this kind of analysis with small amounts on the microgram scale, and to analyze even more chitin deastases, or even using different chitosans from natural sources, and to see if the pattern of acetylation is also non-random, or maybe blockwise or regular or whatever. So to sum up my presentation, first we use this quantitative cross analysis to characterize the products of a lot of different chitinolytic enzymes, like the chitinosanase, KB, TVK, CSNMN, and so on, to understand how these enzymes are degrading the chitosan polymer. And then in the next step, we selected enzymes that are most interesting for this kind of analysis to perform some enzymatic mass spectrometric fingerprinting of polymers. And then by analyzing the products of these analyses, we were able to determine the degree of acetylation, and also get more information about the pattern of acetylation. For the degree of acetylation, we got this fixed number, but for the pattern of acetylation, a lot of other experiments have to be done to get the full pattern. And well, I want to thank all of you for your attention, and also a lot of these people. The first picture is now already 2 years old, but a lot of them are included in this work, because for all of these analyses you need a lot of different enzymes, you need a lot of different analytical tools, you have to have this different chitosan polymers to combine all of these together to get all the results which I showed you in this presentation. Thank you for your attention.
Yeah, thank you so much, Stefan. Really exciting. It's a highly sophisticated method to analyze the PA. Thanks for your contribution. Are there any questions for Stefan? Vincent, I think that's a clapping, right? That's, yeah. I'm still not familiar with the smile.
Clapping, yeah, clapping.
Maybe I can kick off with a question, and already direct that into what you just mentioned, that there are so many enzymes to be produced, which of course takes effort, but it also adds up a lot of information about enzyme specificities, right? Of chitosanases, chitonases, and so on. And I'm wondering, you're producing this tremendously large enzymatic toolbox, which makes a lot of information, structural and other information, biochemical information, accessible. So are you working on projects where you use this information and actually feed in silico tools like machine learning and whatever to actually also predict new activities or predict activities that you want to actually aid the search for new enzymes? Is that something you're also working on in the lab, maybe?
Yes, we have it like, I don't know if it's here, several people working on chitin deacetalases. They're like doing this a lot of in silico work to have we have different mutation libraries for chitin deacetalases, for chitosanases, where we checked how different amino acids are influencing the specificity at different parts in the active site. We could also show, I think, show that some of these amino acids are very important. Yeah. And we try to manipulate the subsite specificity to generate new products.
Yeah, that's really exciting, because at some point I think, especially when I think about research done at de novo protein synthesis in Washington, for example, in the Baker lab, I could really see that, like, if this toolbox is enlarged and more information is getting available, I think at some point, I wonder at which time the point comes where we can just design the enzymes we want to, and in your case, maybe the chitosanases that you want to see and want to see in your studies. Cool, thank you so much. Are there any other questions? I think we're already past noon too, so everyone is looking forward to a lunch. Yeah, thank you so much to you, Stefan. It was a really exciting presentation. Also, thanks again to Thomas. I think it was really exciting to see all your contributions and the research. And yeah, we'll see each other again in July, 27th of July, kicking off with a presentation of Francisco Oquigela. I'm really looking forward to that. So see you next month then. Have a good day.
Thank you, Gregor.
Bye.
Bye.
Bye.